Welcome to the April Hidden Vale Wildlife Centre Lunch and Learn, where today we are joined by Dr Edward Narayan, who's the Senior Lecturer, Stress Research Group Leader from the University of Queensland. So welcome, Dr Narayan.
So
Dr Narayan, he obviously, round of applause. I never get that. Graduated with a PhD from the University of the South Pacific. He's worked on non-invasive reproductive stress and endocrinology tools for amphibians. Edward leads an innovative research program based on the conservation physiology theme. His research platform is based on comparative physiology research that contributes new knowledge on key areas such as animal health and welfare and conservation biology. Edward's research team has made significant new discoveries, including understanding the subclinical physiological impacts of the debilitating pathogenic disease chytridiomycosis. Is that about it?
BDM.
Okay. On amphibians, the physiological impacts and fitness consequences of acute and chronic environmental stresses on amphibians and small mammals. And today, Dr Narayan is going to talk on the applications of conservation physiology theme in wildlife research. This talk will be based on the central theme of conservation physiology, which shaped his early research career based on the reproductive and stress endocrinology and captive breeding of endangered amphibian species. Everyone, please welcome Dr Edward Narayan.
Thank you very much, and I thank the dedicated team at Hidden Vale, Robbie and the team members for giving me this opportunity. So, like the introduction, we are the Stress Lab, and today I'm going to share a bit of the journey that I took through my PhD and then early postdoctoral research and to where I am now at UQ. So the talk will be roughly 40-45 minutes, and I'll touch base on the amphibians and some examples of mammals and marsupials, koalas mainly, to show how we've been able to marry physiology with ecological research issues. But before I do that, there's a lot of people who I've come across and have touched my heart and my academic journey. So it's very much a mateship and getting to know everyone here in Australia and also internationally. So lots of people who have helped me, and I'm really grateful to everyone. Because the audience includes young scholars, so just to motivate them, hopefully there's a 2 cents worth here. Just the journey I took all the way from Fiji with frogs, and then came to Australia, very lucky to be here and being mentored by really top-notch professors, and then went briefly to India to do a postdoc over there, and then did a round trip to Saskatchewan, stayed there for a while, and then came back to Australia. This is home for us and for me and my young family. Got a lecturership, and then with Western Sydney University, and now at UQ. So things are happening, and lots of things are happening from teaching and research wise. So I'm also teaching quite intensively throughout the year. So very lucky that I can actually talk a bit about research for a change. So, we know that a lot is happening to biodiversity, and it's very challenging times for native wildlife conservation, and the challenges are all the way from grassroots level, all the way into the, in Canberra. So the discussions are very robust, and we know that, a lot more needs to be done than what is being done right now. So it's a very complex, interaction between wildlife conservation and the challenges that are being faced. There's some examples here, the things that you guys maybe know and know a bit more than what I know. And you can see, there's underlying physiological changes that actually impact on the ecological, fitness of your study species, which is pretty much really important for us to understand. And it goes hand in hand with ecology. So my group, we believe in the stress triangle. So basically, we are trying to look at the interactions between host physiology and environmental trauma, and some disease, for example, BD, and being able to look at some model species. So conservation physiology was coined within the last decade, and it sort of became pretty much aware that we need to apply more and more of endocrinology, genetics. A lot of work was already going on, veterinary medicine, and it's actually a part of that big conservation puzzle. And when you're looking at conserving really iconic, rare, cryptic species, a lot of, you'll see that with my PhD work, luck was on my side as well. And these mechanisms are actually, it's like a fulcrum of interaction between various factors, and if you are aware enough, you will be at the right place and be able to spot that really significant data. So the journal does exist. I'm on the editorial board of Conservation Physiology as well, and it's a really good opportunity to show some of the physiological tools available out there. And just recently, my current PhD student, Harsh Pahuja, who is in the crowd, just raise your hand, there he is. We managed to cook this paper. It was took more than a year. I met Harsh and we were like, we need to get something out there. And it was like during COVID, I had lost it. And luckily, I met some dedicated students. And this is just one example of the small achievements that we have been making. So this is like using a physiological model to look at how lizards are responding to environmental change in various landscapes. So my thing is all about stress. I talk a lot about stress, and I'm trying to understand, just having a yarn with Vea. Before I was saying that I want to know more and more about how the stress physiology operates in animals. And going all the way to the father of stress physiology, Hans Saal, he said adaptability and resilience to stress are the fundamental prerequisites of life, and every organ and function participates to. So as long as you have a beating heart, you still have a chance. So you might recognize a few faces in those pictures because the world is so small, we are so interconnected, and talking to some of the colleagues here, it's just amazing where we are seated right now. It's just brilliant. So, the whole thing around conservation physiology, every animal, I put a frog in there, there's no bias as such, but it's basically the interconnection between the external environment and the intrinsic physiology of the animal. So for example, cortisol is one of the biomarkers that actually helps the animal to overcome stress. So it's not a bad thing. As long as there's some stress, it's not a bad thing. It gives you that opportunity to respond to a particular situation. So you have this thing called an acute stress, which is basically your emergency life history state. And things are on resting homeostasis. So if you have a predator-prey interaction, you might see something like that. So that basically is a metabolically demanding hormone. So it will actually help you to move your actin and myosin filaments to allow for the muscle activity to occur. A good example of acute stress, the work that I did a few years ago with Vea Nicholson is in the crowd as well, and you can see the work we did on the bilbies just to show in 3 males how cortisol levels were highly dependent on the individuals themselves and the extrinsic factors. So a healthy control male, low levels of fecal cortisol metabolites, and this guy was being taken to a school visit. But this bloke here, he was one of the hot male, so he was highly used for educational displays. But you can see that there's underlying haywire of physiological response when he got injured. So it was just an example of an acute stress response. And once the stressor moves away, then you can see it goes back to happy days. But that's not what I am interested in now. Look, I've done some work with zoos early on during my postdoc. I've moved my attention away from that because I want to understand how animals are operating in the real, war zone of stress, which is in the wild, and and being able to understand how we can measure the spectrum of stress, and if we can actually provide appropriate interventions to then reverse that stress, because from the biomedical literature, it's already known that you can regenerate the dendrites and healthy, if you put enough water and for a long time, then the trees will start to fruit again. And so a very good example of chronic stress is of bile farming. And This is a work that I am currently collaborated with Animals Asia. So these guys are kept in very horrific environmental settings. And they are brought back into the sanctuary for a safe haven. So these guys are never going to be reintroduced back into the wild. So there's a lot of conflict with regards to wildlife conservation and the use of bears in traditional Chinese medicine. And Animals Asia is doing a lot of hard work in bringing them. So over the last 10 years, around 700-800 bears have been rehabilitated, and they are building a new facility just now. And so being able to work with the bear team in Vietnam, we did some study to demonstrate when the bears were being rescued from the farm and then brought back into the sanctuary over 22 weeks. It's not a fairy tale story. There's a lot of integration required. Some bears will never return to normalcy, but it did show here that with this simple measure, it's a powerful technique in this case, being able to demonstrate how stress levels are changing through time. And it provides a bit of an animal welfare dimension to what you are doing to your animals as well. So we see that there is a lot of individual variation in the stress response of animals, and it is something that I'm also very much interested in. But when you see animals which, for example, are showing no signs of, like cortisol readings are on the floor, and you might say that, yep, that's good as, but that's not the case. Here we can actually see that stereotypy, over the roof. I'm looking at Popeye at the moment, trying to understand why his cortisol levels were on the floor, but you can see he was showing, stereotypy, and then you've got a barrage of, stereotypy and things that, like vomiting teeth chatter. It's like really awkward and really sickening to see, and you can see that health issues are also pretty much high in these animals. And so just showing the bilby as your example, I know I'll go back to the bilby as well with regards to the wilderness, but that acute stress in a captive situation to a real example of what chronic stress is, maybe we can think a bit about here in Australia. There are some examples of that. We've tried to advocate for stress in bears and being able to talk to the people and put your ideas out there. Just last night, while updating my presentation, I bumped into this paper, which is an analogy to what's with the bears, but this is a naturalistic situation, maybe rapidly occurring with climate change, with the researchers are actually saying that the polar bears have signs of chronic stress in the blood. And they saw some really cool physiological changes with regards to the HPA axis, so something for us to have a look up on later. And when you look at this, again, it's just showing you the pathway of the stress response. And then eventually the cortisol will, or the glucocorticoids will bind to the receptors and then go and promote your behavior to support, blood supply of glucose. Or it can actually, in long term, inhibit your growth and development, reproductive activity and immune function as well. Like when you talk about immune function, I think for animals that are being rescued and brought into the clinic, it's really important, or even wild animals, it's important to understand the spectrum of stress response so that you can see where the stress levels are sitting, and you can understand that if you combine that with your immune markers. So going to my origin, basically ground frogs, my supervisor asked me, Edward, have you seen a frog scat? So I said, yeah, I have seen it. Can you collect it for me for the next 3 years? So what I did was I started off going by boat to this island every month, collecting frog pee. And it was basically to validate this non-invasive method that we can then use to understand what's going on with this species that's only found in this narrow zone of environmental habitat. So there's only 60 people on this island. It's a lovely tiny island, main island over here where I was born, and take the boat, go and spend the night there catching frogs. When I first saw the frogs, I was like in Mecca, like lots of frogs everywhere. I became crazy. but the island is gridded, so every small section belongs to a different family. So I had to live with, I met, all the community people, and all the houses look same. So I ended up in wrong houses as well at sometimes at like 3 a.m. in the morning, coming back from the bush. The journey was to understand the ground frogs. In New Zealand, they were setting up endocrinology techniques with the Archie's frog. As you know, this is one of the other highly iconic and evolutionarily important species. And so I was lucky to be involved in the ground frog conservation project. And in New Zealand, the Ecogene Lab in Auckland, they were setting up with the Archie's frog. So my job was to look at the endocrinology of the frog on the island, and then also bring some frogs with me. Paddy Ryan, a naturalist, had seen ground frog eggs. He sort of knew what it looked like. My job was to go and find these eggs in this island, get the frogs, breed them. That's my PhD. So, okay. So miracles do happen, and it did happen. The interesting stuff that I will share today is with the hormone monitoring work. I could have submitted a PhD thesis with just the ecology stuff, but then after 2.5 years, I moved to New Zealand with all the urine samples, being stopped at the airport, what the heck are you carrying with you from Fiji? 2,000 frog pee, what is this all about? So, but those are the things that you'll expect. And it was really interesting, some of the things, looking at the developmental biology of the frog itself, which is really cool. And being able to draw, the actual developmental biology. These guys actually don't have a tadpole stage, so they lay eggs like birds in a nest, and they would actually, a froglet would jump out after 30 days or so, which was really cool. And you can see that using a candling technique, I was able to look at the underbelly eggs of the frogs, and being able to categorize, because in Fiji, it's full of rain, and it's like, a rainy island. So what is outside breeding, what is within breeding? So I needed to sort of justify what's the active breeding phase by looking at the size of the underbelly eggs or the oocytes itself. If they were greater than a certain mass, then they were actually actively breeding. So that's me in New Zealand, and setting up my assays. And so with the frog urine samples that are collected, we had to validate it against each of the antibodies that you have targeted it for. So for example, you can measure progesterone in your females, estrogen conjugate in the females, and males testosterone as well. And what you can see over here, there's virtually nothing known about this frog. So by using frog pee, we were able to tap into the biology of this endangered species. And you can see that with regards to longitudinal hormonal profiling with the male frogs at least, each data represents my trip to the island, each dot. So you can see over there with the males, high level of testosterone and intra-individual variation during the wet breeding season. And this is back on the campus of the University of the South Pacific, where I brought a small piece of the island with me. You can't create an artificial cage and assume that the frogs will breed. So at Koala Park, they had some frogs and they bred. I ran to Koala Park, brought the eggs, and later a gecko came out. So my supervisor said, "You've saved another endangered species by accident." I said, "Okay, that's really nice to know, but that's not going to help me with my PhD." So getting the frogs in, these frogs are very funny. They actually smell the landscape. They have a bit of a weird interaction with crabs. They are sort of saying that it came from South Africa. There's a bit of a, the salty crab frog, crab-eating frog in South Africa, which is really cool. And so, how do you breed these guys? Maybe by chance I had got some gravid females with me. We did not do any artificial insemination as such or try to stimulate their breeding potential. But what we can see over here with urine sampling, we were able to show whether the female will breed or not. Because, for example, at Koala Park, they used to check the frog every second week. And what would happen is that the frogs would drop their eggs mid-development. So that is then basically a breeding failure. What we then found out was that the same information was observed in the hormonal profiles of the female, because progesterone, if you look at the the unshaded squares, progesterone in these two females, progesterone is what helps you to continue your pregnancy through to the late stage. And this is what we can see over here, that in female number 1, who eventually ended up breeding, had progesterone cycle in combination with estrogen conjugate. Female number 2 basically did not have that. So that's, the story of the ground frogs, getting the froglets. The other funny thing is that the froglets look very similar to the cane toad froglets. So how the heck do you do conservation management with the invasive cane toads? The cool thing is that the ground frog froglets have this butterfly pattern on their back, which we are saying that we can use that to distinguish between, because the cane toads would sit on the nests of the ground frogs waiting them to hatch. You've got a whole life cycle and a whole extinction crisis in front of your eyes. So it's a very complex topic to deal with, and this is just me in my happy days, looking at the froglets when they were born. For the first time. And you can sex the frogs as well. So you can look at certain hormones like estrogen conjugate, which is found in the female sex. You can see that in males they are pretty much low, and then females which are carrying eggs, they're a bit high. And then because these are monomorphic species, so you can't tell apart whether it's a male or a female. So, you can have a bit of a problem with trying to initiate breeding. Testosterone cycle of animals in the wild. You can see quite beautiful patterns over there. And estrogen and progesterone. Just look at the bars. I would not be too deep into this, but that's pretty cool pattern with the Fijian wet season, which is really cool. And then in the last 10 years, I was like, okay, Edward, I moved to Australia, and I was just thinking about how can I give back something to Fiji, because that's where my roots are. I have been collaborating with colleagues in the US to come up with a bit of a network of information to carry home that we can use conservation physiology techniques. So, for example, Caitlin Gabo's lab looks at salamanders, and Zach, he did his PhD, and I was one of the co-supervisors looking at the effect of artificial light at night on the endocrinology and developmental biology of frogs. And Drew also does lots of cool stuff on frogs in the US. So that interconnection is very important as well, to be able to network with other colleagues who have the same idea and the same techniques. So Gabo's lab was the first one to actually measure hormones in water. So they could actually retrieve water and like environmental DNA and measure hormones in that. Then luckily, being an Fijian Australian, I got the Australia-India early career fellowship to go to India. So, okay, let's go to India. So this is a lab that I was collaborated with in Pune. And that's one of the mountain chicken, a big frog, which you have to catch. It's very fast. You can see over here. And so using the Bombay night frog, it's a very funky name. This is a male guarding his eggs. So you can see that he's like, okay. And the same science we are bringing to the different setting. So we are able to validate the methods going to this island, this mountain, to then collect the frog urine and being able to validate the method. For example, you can see over here testosterone, and you can see the patterns, the lines. As long as the lines look like this, happy days. If your lines are like all fuzzy, then your graphs are not good. My students would know. Gina is smiling. And with the biological validation, you can stimulate the HPA axis, or you can stimulate the HCG, HPG axis as well, and be able to stimulate that and show what's the actual physiological response in your animals. So we have done this with like bilbies. I think Vea actually injected the ICTH in the bilbies that we were studying, which is really cool. And the same thing you are able to demonstrate that in the lab. And I think over here, there's 3 frogs. So you can see over there, there's something going on. There's some amplexus, and this guy is just, And so those were some cool observations that I made while being on this exotic landscape. And you can see some images here of the place where we were at. The males would have this underlying impacting features, which would be really cool as well. And then what we did was we basically brought some of this technology. I trained up a student, Amrita Joshi, who just finished a PhD, and basically looked at the calling behavior of frogs and looked at the interactions between testosterone and calling activity in the frogs. Now, coming back to Australia, there was this cool frog, the Litoria wilcoxi, which my Mark Hero, the famous hepatologist who was my postdoctoral supervisor at Griffith, he said, "Edward, I've seen this frog which changes color in seconds." I said, "What?" He said, "Yeah, that's what it does. It becomes bright yellow during the breeding season. What's the reason for that?" So we became a good partner as an ecologist and the physiologist. I said, "I can measure hormones in pee." He said, "Okay, let's go to the bush, collect frog pee." So Victor had come from Brazil that night. He dragged his suitcase into the forest. John Shuka was also with us. We are basically putting frogs in containers and looking at their color scores and collecting their pee to look at the testosterone and corticosterone profiles. So this is what it basically looks like. Hopefully the video works. Maybe it's... So what basically that video was going to show was how we created a potion, a magic potion, and it was epinephrine. So we put this epinephrine onto the skin of this male frog, and you can see it changes color from brown to yellow within less than a minute. And what that actually told us was that why would you want a frog to be brightly yellowed in the middle of sex, basically? That's, inviting all the predators. So there's a bit of an ecological trade-off with the need to breed and continue your next generation, and being able to have this sexual dimorphism, which was pretty cool as part of Christina's PhD project. Thanks. So what we then discovered was that there are these chromatophores on the skin of the frog, which actually is activated in response to this sexually, hormonally driven ecologies, which was really an answer to the big question that Mark was trying to find the answer to for the last 30 years. And I was a small part of that big discovery. So what I'll do next is talk a bit about koalas. Hopefully I've got a bit more time, 15 minutes maybe? Yep, cool, thanks. I don't want to leave this place without talking about koalas because this is the koala hub, so. Yeah. It's just when you have so many things to talk about, you are like lost as to what's the best thing to say. So hopefully it's all coming together, but just drag it halfway through. Thanks so much for your help. We've done the polar bear, we've done the frogs, slowly, yep. Frog breeding cycle. So that's the journey with the frogs, and then color change, yep, so stop over there, that's it. So then I was finishing my PhD and Mark was examining my thesis, and I met Daryl Jones in a OMS conference in Perth. At lunch he said, "Are you the guy from Fiji?" I said, "Yeah. We have a Center for Innovative Conservation Strategies. We want you there, because we are trying to save the Kurumbin tinga frog at Kurumbin Wildlife Sanctuary, and we'd like to see the endocrinology of this." I said, "Okay." So I applied for the Griffith postdoctoral fellowship, and I got it. So Mark ended up sending me an email saying, "Edward, your thesis was amazing. I'm so happy that you've done this work, and come and work with us." So packed my bag, said to my mom and dad, I'm going. And that's where my journey with, where I am standing right now started. So taking that technology, I could have, stayed on with frogs. I said, let's look at other, because I was in love with all this cute, this Australian landscape and the animals. And so taking that techniques with me, the little knowledge that I had, to understanding some really complex topics, and some of this is actually beyond this talk itself. So hopefully it will make some sense. So with the koalas, we all know that. I don't need to repeat what's going on. And so my interest right now is how can I work with the koala, rescue groups or wildlife, warriors, to then provide some more understanding of, of the physiology of these animals before time goes beyond me. So it's really important to get as much data as we can get to save the conservation conundrum. So how much stress can the koala bear? We know that that's what's happening to it. I'm really interested in understanding the dynamics of the HP axis, in understanding how the koalas are responding to stress, and the spectrum of the stress response, how we are moving from an acute state to a chronic state. And what does it mean when you talk about fitness of an animal? I think those are some really interesting questions. So starting off from the basics, with access to dream world and Bear's wisdom and support, we did some work. Christina was an honor student then, and basically looked at the dynamics of stress responsiveness in male koalas and female koalas, and you can see that male koalas had a bit higher cortisol levels than females, which were actually carrying pouch young. So there is a within and between individual variation in these animals as well, which is something to keep in mind. And there was a bit of a discussion whether the koalas actually are able to recover from stress or not. We know there's so many environmental trauma and the disease itself is really damaging these animals. So this is how you would tell whether the koala is stressed or not. It's basically looking at the adrenal gland. So these guys would have modulation of the adrenal gland, and this is a bulged up adrenal gland of a dead koala basically. So this was Nicola's work. I was talking to Greg earlier on, and there's been some really cool research that have demonstrated how you can apply physiology to a very important ecological question. And other colleagues have also looked at stress in koalas. So it's still an ongoing topic of discussion. But we know that stress can actually lead to damaging effects on the reproductive biology, immune function, growth and development of your animals. So what we basically did was, my job was to validate a technique to measure the glucocorticoids in koalas from scats. So to do that, we actually did a biological validation by collecting blood samples. You can see plasma cortisol levels all over the place with the individual in the blood. And it's actually like a minute scale response. So you have to be, these guys were under anesthesia, I guess, but with wild animals, they would be highly stressed. And so that's why we want to use non-invasive sampling. What we discovered was, I went back to the lab 10 times, 20 times, I looked at the data, I can still visualize sitting next to Mark and looking at this data. And he was saying, why the heck do these koalas have 2 cycles of physiological response to your ACTH challenge? It's because of the complex gut system that these guys have. They are not like your sheep or chicken. Okay, sorry, Gina. So you can see the first question we were trying to ask was, how stressed are these koalas? So this is some data that I'm going to show very quickly with the work that I did with one of my students in South Australia to look at the dynamics of stress with habitat and environmental stresses. We know what's happening with the population effect and urban to rural fringe. But what I'm more interested in is how can we desensitize the stress response so that when animals come into the clinic, you have them a better chance of recovering, because chronic stress can have very self-damaging effects on the the psyche of the animals and the health as well. and the recovery. So these are some koalas from South Australia, and you can see that they've been brought. These are actually sick and injured animals. They are not like wild koalas, sort of like they were being, and so that's one of our students who was actually in the lab collecting the samples and doing all of the work, and we validated the methods, and just looking at the dynamics of where the koalas are, they move quite a lot, as you would know, and they wouldn't be staying in one place. So there's a bit of a temporal variation in terms of how the stress response would change. And I think the work that Renee is doing at the moment would hopefully open up that picture as well as to what these koalas here have been up to. But what we can see here, we are working with a lot of data, and we are getting a lot of clinical data on koalas. And so, we are trying to sort of sieve apart a lot of clinical data. Some of them actually have diabetes and a mix of, stressors. And we're looking at their blood profiles, and then what sort of treatments are being given to these animals, and then eventually what's the outcome. So I'll show you some examples here. For example, this red line is I don't know the word happy is the right word, but like healthy koalas, a little subpopulation in a forest around Brisbane somewhere, and you can see that their stress levels are pretty much on the ground. Nutsi came into the clinic. This is the South Australian koala. You can see the stress levels are pretty high. What happened to this koala was that it was actually given prednisolone, which is a corticosteroid derivative, to overcome certain clinical issues. And eventually it was diagnosed with the diabetes. So there could be an interaction between cortisol being infused into animals and the clinical outcome as well, which was pretty new to us. You can see some animals come in with a lot of pain. They're unable to walk. The stress levels are pretty high in these animals. So maybe this could be a predictive tool to then be used with the clinical data, which is ever more important, to then understand what's happening with these koalas. And you can see the HPA axis was actually exaggerated and activated with cortisol, causing widespread dysfunction with pain as well. Nigel, there's another koala here which was found on the ground drooling, eventually euthanized. Stress levels are very high. So what you see, these boxes here, they are telling you the lag time. So what you are measuring here in the first 10 days at least, that's like the window of stress that the koalas have come in with the clinic. So there's a lag time between how much stress is actually being carried on over because of the metabolism of the stress levels themselves. It was not all a bad story. We could see that, like the work we did with the Asiatic bears, we were able to demonstrate that stress levels were slowly starting to come down. The animals were being given with antibiotic treatment, with certain interventions, good rehabilitation techniques, and they showed some good improvement. And there were some happy koalas with negative chlamydia released with the Kera. What eventually happens to these koalas, we are actually now tapping into that story with Harsh, who's doing his PhD on the koala joeys. And you can see we are able to look at if a koala comes with a dog attack or with a disease, how the stress levels are being varied all throughout the clinic, before and after story, which is not a fairy tale story, but it is part of the big picture. So we saw that a lot of koalas were being euthanized and less were being released, which is something we all know about. And the more the animals came with chronic conditions, like physical injuries or some certain infections and stuff, the more stress levels they carried. And for example, animals with mild conditions grew in a good improvement in veterinary care, tagged and released Margaret. You can see her stress levels were. So at an absolute level, we know that there's a difference between males and females, but this is more responsive, I would say, more responsive to the extrinsic factor than the internal biology, because now we are behind the stressor. So it's like the physiology is lagging behind. So it's whatever I'm seeing is actually the effect of the environment more than anything else. So how much stress can a koala bear? Hopefully this video will play, but I don't know if it's as important right now.
New research suggests koalas rescued from rural areas stressed than those from urban centres. It's believed to be because the native animals from more populated areas are accustomed to having people around. This koala, Joey, is having a final checkup. She's got her bling on. It'll soon be released back into the wild. It's one of hundreds treated at the Port Macquarie Koala Hospital every year.
Apart from disease, which is the main reason for admission, we see koalas hit by cars and unfortunately attacked by dogs.
When it comes to safe and injured koalas, the animal's stress level can be a deciding factor.
So it can have a major impact on their treatment.
A new study from Western Sydney University has measured and compared stress levels in koalas rescued from urban areas with those found in rural settings.
We are trying to understand how they respond to environmental change.
Edward Narayan and his team used scat samples sent in from wildlife groups to detect stress hormones.
We found that the koalas actually that live in rural areas, in rural to urban fringe, were the most stressed. What was really interesting to us was that koalas in urban areas were actually showing lower signs of stress.
The researchers say the findings don't mean koalas in urban environments aren't stressed. It simply means they're less stressed and appear to have become better at coming into contact with humans. They believe it's evidence the animals can adapt to having us around, but they need to have protected habitats.
If we don't deal with stress with regards to our native wildlife, we will see more and more beautiful animals will end up becoming extinct.
Something no one wants to see. Jonathan Hare, ABC News, Sydney.
So that's like, it was part of, that bigger picture that already exists. And so I was given this task with a NGO who gave us some funding to go and discover how much stress these koalas are in a landscape, which is undergoing massive change. So I took my bag, hit the plane to Ballina, and was collecting koala scats in a bit of a rugged, like bit of a rogue setting where they said we could have people shoot us if they saw us. So what we did was I ran a marathon of, because over these years collecting all these scat levels from koalas, I'm starting to see a picture. It's not an artifact of my blurred vision. It's actually a true biological thing. And you can see that what I did was I ran a marathon to, provide a bit of a discussion. It's a very complex topic. But what we saw with these koalas that were undergoing heavy land clearance, their stress levels were quite high. And, you can see chlamydia was sort of a bit far behind, and bushfire was also next to it. These are healthy wild koalas. So I'm interested in seeing more and more koalas over here. So the collaboration, hopefully, with what we are doing with my PhD students, and we can get some more data and be able to add to that bigger picture and be able to contribute to that science. And now what we have also done, and that's some of Renee's work. You can see she published this with me for her master's, and we were now able to, that's not Renee, that's Dylan, who came for the photo shoot because Renee was in the field. So no credit to Dylan, he's measuring plant root at the moment. So, but, which is also a good thing. But what we can see over here is that this is hair. So we can collect hair samples and measure the hormones in the hair itself. So, there will be always two sides to the team, but this is a pretty powerful technology as well, which is available to us now. And hopefully one day Renee will be standing here and giving a talk on her work. And so, we've done work, and that's where I'm sort of headed to try to understand how animals recover from stress. Stephanie Hing, my PhD student, with the colleagues from Murdoch University, said very clearly that it's the underlying stress. We can have debates about these big picture topics, but while we have time, I think we can, sort out this sort of stuff. Still will be required to transport animals. We'll have issues to deal with regards to social stress, animals in the wild. So with Violis, we did some work as well to show the seasonal variation in stress response. Just wrapping up my work, and you can see over here with bilbies, all the way from, zoo zoos into the semi-wild and onto the wild. You can see. So I'm really interested in working with field ecologists. I don't call myself a field ecologist anymore. I'm more of an integrative physiologist. But you can see that we can see animals who are, eventually cats entered Caruinia, and you can see what happened. There's a high intra and interspecies competition with rabbits and all sorts of, furry stuff over there. There needs to be more work, and hopefully we'll have more collaboration in these areas. With animals where you can't inject ACTH to biologically validate things, we've actually shown that we can look at simple interventions like collecting milk, which did show us stress. By stress, it doesn't mean that the animal is about to die. It's just what the word is. It's the stress response. And that's Giles and his team. They are collecting at Australia Zoo. So we were then able to look at, and the researchers, the managers were very happy to include, we're not saying animals are stressed, that's what their biology is. And so it's really important to understand the dynamics of stress in relation to hands-on, hands-off, handling techniques of animals and the environment that they're being provisioned with. So that's where I will finish my talk. Thank you so much for your time. I'm sorry I went a bit over. A lot of people to thank. My team members, especially Gina, Harsh and Ranay, who are the Stress Lab senior members. They have been with me for few years now, and so I'm really looking forward to their PhD works. And yeah, thank you, all of you. I'll leave you with this lovely picture of a Joey young koala eating leaves. Thanks.